morning, everyone, and welcome to Kansas City Oasis. We are one of the alternatives to faith-based community that exists here in Kansas City area. We gather around our values, which include meaning comes from making a difference, reality is not the reason, be accepted, human hands solve human problems, and maybe most importantly is people are more important than beliefs. We're so fortunate to have with us today a musical guest, Mark Pagano, who's going to sing for us. Our community moment today will be from Gary Blakely, talking about his adventures in South America recently. And our guest speaker will be Katie Griffin Hill, and she'll be talking about preclinical to clinical biomedical timelines. So we're really pleased that you have chosen to join us via Zoom and on Facebook Live, because we really feel like, especially now, the human experience is better when it's shared. And we're really pleased that you have decided to spend this time with us this morning. Just one announcement briefly before we get started. We still have some opportunities to submit button ideas if you're interested. And those again are focused around our 5 values. So if you go to the Facebook private page, you can see the details of how you can submit your button ideas, and we'll produce those. And hopefully once we get back to meeting the community and being out in public and tabling, those will be one of the things that we can do to generate interest in our community. So we're really looking forward to those submissions. So without any further ado, I'm going to turn it over to our musical guest for today, Mark Pagano.
Thank you. Can you hear me okay? Okay, good. Well, thanks so much for having me. I'm Mark Pagano. I'm with a band called Fire Dog in St. Louis. And we were supposed to be in Kansas City this weekend playing at the Kansas City Folk Festival, which has been postponed until maybe next year. So I'm broadcasting here from my home to you, my natural habitat, and I'm going to play a song from one of our albums called Endangered Species Project. Hope you all are enjoying where you live. We've been spending a lot of time around here, and we're doing just fine. So thank you so much for having me. Like I said, that song is from an album that we put out a couple years ago called Endangered Species Project.
Cool.
And it's kind of an educational concept album meant for young audiences. I'm a teaching artist also, so I go into schools and do songwriting workshops with young people. I was going to play a song that was written with a 4th grade class. You can find this song on our album that just came out in the fall called It's Fire Dog. And this song is about an animal, kind of a mythical, well, it's a real animal, but it's the narwhal, unicorn of the sea. And if you don't know anything about this animal, you might learn something. Narwhal, unicorn of the sea. It can grow between 13 and 20 feet. Polar bear and orca are its predators, found in the Atlantic and Russian waters. As big as a bus, narwhals swim free, a cross between a unicorn and a manatee. These large mammals eat fish for food. Their tusk is just an overgrown tooth. Narwhal, narwhal, unicorn of the sea, the sea, the sea, the sea, narwhal. Loving those unicorn horns. Their babies are born blue and gray. They change colors as they age. They use their tusk for echolocation when they're searching for some tasty crustacean. Their tusk has 10 million nerves. When they swim in curves. They live to be 30 or 40 years old. One lived to be 115. So we've been told. Narwhal, narwhal, unicorn of the sea, the sea, the sea, the sea. Narwhal, narwhal, unicorn of the sea, the sea, the sea, the sea. Narwhal, 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 unicorn of the sea. Narwhal, unicorn of the sea. Unicorn of the sea. Narwhal, unicorn of the sea, the sea, the sea, the sea. Narwhal.
Woo, woo. I'm the only one you can hear, so I'll do all the clapping.
Thank you so much.
Thank you, thank you. That was fun. Narwhal. Fantastic. So we're gonna, right before we introduce our community moment speaker, we're gonna talk a little bit about some celebrations that some of our community have shared with us. So first of all, I'd like to wish Lynn Thompson a happy birthday. Her birthday was this past Thursday. And then Mayan Ruins, her birthday is coming up this coming Friday. So that's very happy birthday to her. It is their 11th anniversary, so it's great. And then also you guys should share in our testimony that Rachel graduated and we're both international on site, so I'm very there. So we can share our celebrations and already say that is that he just had recently. And if, oh, my audio is cutting out. So he is subscribed as news methodical and procrastinator. And if you don't here, so go ahead and your presentation.
Should I go?
Yeah, sorry. I don't know if you can not hear me.
We lost your audio. Your audio wasn't working at all. So
Okay.
no one has heard the warning label or anything.
Okay,
but,
hang on just a second. Just give me
anyway.
1 second. Let me, here, hang on a second.
Everyone give me a thumbs up if you can hear me. Thumbs up if it sounds okay. Okay, good. Then I'll just start my presentation. Now, this is a short presentation, very rapid fire presentation of my trip to Central and South America by motorcycle. We started, I left Kansas City with 31,920 miles on my motorcycle. I put almost 50,000 by the time we were done. We did have to kind of rush through Central America to make a appointment with a boat that was going to take us from there to Colombia. There's my motorcycle loaded down with saddlebags and rain suit and an emergency tent. We didn't plan on doing any camping, but you need to kind of be prepared for that. Basically, it's a big dirt bike is what it really is. It's street legal. It'll handle the highways, but it's also kind of just a gigantic dirt bike. This is an example of the kind of roads we were dealing with there. The Andes are very steep and the roads have to do a lot of snaking in places to get across tops of mountains. So that obviously motorcyclists love that kind of a road. As I said, we rushed down to Panama through Central America, but if we could catch something close by, we did. This is Tenosique in Mexico. It's some of the Aztec or Mayan ruins, and I have a trouble keeping up with Aztec and Mayan and Inca and all that stuff. But yeah, ruins going back to pre-Columbian times. When I say pre-Columbian, the arrival of Christopher Columbus. The next slide is hotel amenities. What you see here is when it gets there is the typical soap, shampoo, and a condom. We got a condom in the hotel. Why do we have that? Well, the nicest hotels in Central and South America are what they call auto hotels. And the reason they call them auto hotels is a couple goes or someone goes for a little tryst and they can drive into a garage and shut the garage door and have total privacy, and no one can even see them as they're fooling. They won't even be able to drive through the parking lot and figure out who's inside there. Hotels were quite an experience on this trip. The next slide coming up is what's called a suicide shower. When you go in and you're asking about accommodations, you ask about, in our case, we need a room with 2 beds and we want to know if they have hot water. Well, a lot of the hotels were never plumbed for having hot water, and this is the solution. They put in a heater that's right on the end. It is kind of scary looking if you really think about it, the wiring coming right there. This is an example of some of the kind of roads we had to deal with. In fact, this was one of the worst sections that we had to ride. And this was actually the road recommended by Google Maps, believe it or not, to cross from Guatemala into Honduras. There you see the border, and the end point is La Entrada. See, it says it should take 1 hour and 53 minutes. Took us about 6 hours, and some locals had to help us and had to give us a lot of help. And so there we were in the part of the world that everyone thinks we're going to get our brains blown out by, bandits, and locals helped us through this section. We'd have been in real trouble if they hadn't done that. But we're kind of out in the country, but yeah, that was, Google Maps let us down there. Otherwise it worked fairly well for us. This is a gas station. See the gas station attendant and his gun. The security situation down in that part of the world is pretty serious, in Central America in particular. We saw this more than once. But he's in uniform, and we never actually had a bad experience. Now, you also heard people talk about not drinking the water. We avoided it when we could, but you can actually buy bags of purified water. That is a plastic bag with good water in it. Has no way of opening it. You just bite through a corner of it and have a drink. That's kind of interesting. And they came in various sizes. This is a shot from a border crossing, but I believe it was into Costa Rica. What you're looking at there is actually an electric Harley Davidson. Harley Davidson was doing a promotion, and they brought two motorcycles down to the bottom of South America. So they were riding in the other direction we were going. The two riders on the electric motorcycles were Tom Borman and Ewan McGregor of Star Wars fame. I got to shake both their hands, although at the time I didn't know who was who because they had helmets on. But again, this is a promotional, from Livewire. It's a Harley Davidson Livewire. Thank you for the commentary there. But yeah, border crossings were kind of interesting. I have this picture in here because chicken restaurants are just everywhere in Central and South America. They eat a lot of chicken down there. And I just was quite amused that they have these happy chickens here. I'm thinking, yeah, you're gonna, these happy chickens are smiling. You're about to cut our heads off and eat us. I just thought that was kind of strange, but that's pretty typical. This next shot is we finally have made it to the Panama Canal. So that is the Panama Canal locks right there. What you see way in the background with smokestacks, or above the smokestacks of that boat, or what looked like smokestacks, that's a bridge that crosses the Panama Canal. I had gone over that because we got to go a little bit further south to meet our transportation into Colombia. You cannot ride, well, you can, but it's really difficult. Below, south of the Panama Canal is what's called the Darian Gap. And technically you can get through there, but it's mainly indigenous people. It's like little narrow trails. So most people take a boat. So that boat you're seeing there is a sailing ship, and it's got, it can be powered by the wind if the winds are favorable. Have I already run out of time? Okay. So they load all these boats on that ship. It's called the Stahlrat. It was built in 1903 and would have been steam powered at the time. This is the kind of view we had riding from Panama to Colombia. The waters look pretty good there. And that boat, little dinghy there, had just delivered up a load of fresh lobster to us, which we had for lunch. Well, in short order, we were in rough seas heading for Colombia, and we all tossed our lobster, including me. It was pretty rough. We have this is kind of the community table they have set up there. There were 17 motorcycles and about 21 motorcyclists on that trip. That's, and the captain there is the guy with the shirt off at the very back of that picture. This is the view of what's coming up here is Cartagena, Columbia. And actually our boat is out there. This is a view from one of the hotels. So we disembarked in Cartagena. It took a little while to get everyone processed through customs and entry and everything. And then we, proceeded from there. Cartagena is a pretty touristed area, as you can see from a lot of high rises and high rise hotels. So this is the town of Minca. We went to Minca next. Minca is kind of an eco-tourist place. I think a lot of the Oasis folks would feel right at home in Minca. Very, very nice place. One of the more jungle-like areas that we went to. This is from Bogota's Gold Museum. It's a gold museum with lots and lots of gold on display, pre-Columbian art and things like that. Really interesting. In the big cities, you can also get some good food. We like this, Motos de la Derecha. That means motos stay to the right. You get free passage on the tollways. That was nice. We like that. Let's far as the food in generally speaking was pretty bad unless you found a place that catered to foreign tourists. Here is a pizza restaurant. Obviously their signage is in English. So they're definitely catering to the foreign tourists. That's where we had to go to get good food. Otherwise, I hate to say it, it was pretty bland and some of the toughest chicken I've ever eaten in my life in some of these places. I took lots and lots of pictures of the landscapes, but as I was going through this, they just don't do justice to it on an iPad or in this kind of a delivery format. But it was just beautiful, gorgeous everywhere, green everywhere, just the most breathtaking landscapes you see. And I just finally had to just stop and said, I can't take a picture of every single one of these. I've taken hundreds and hundreds of pictures, but this is around Finlandia. This is where we stayed around Christmas time at a place called Steel Horse, which caters to both motorcyclists and people who like to ride horses. So hence the name Steel Horse. This is an area called Corcora, which is in the Los Nevados National Park. I believe those are some of the tallest palm trees in the world. And it's a mystery to me how the seeds would ever go up the hill and germinate. It seemed like once they fell down, they would just roll all the way down the hill. But anyway, so the next one is flowers. Flowers everywhere, flowering trees, it's just really beautiful what you get to see there. This is a desert, actually, the Tatacoa Desert. It's the greenest, wettest desert I ever saw, but apparently it dries out some parts of the year, so it can be a desert. Here's some more, some cactus I've never seen before. The next one is just an example. You get a lot of rain and a lot of road washouts, mudslides, rock slides. That was something we had to contend with quite a bit. This is Tulcan, Ecuador. We just crossed in. This is the cemetery. They have topiary artists who carve these bushes. Very interesting. Here's a shot of kind of before and after where they've trimmed up one and they're about to get to the next one. This is me taking a selfie on the equator. Notice how deftly I put my right and left hemispheres in the right and left northern southern hemispheres of the world. The blood was swirling in two directions in my head. It felt fun. I'm kidding. This is Pujili. It's one of the crater lakes in Ecuador. That was pretty cool. I couldn't actually get the whole thing in the shot from where I was standing. I took this picture because they farm and they grow crops everywhere. The side that we're actually looking in the middle of the picture is much steeper than what you see to the left. Just amazing. They just grow food everywhere. Bad hotels. That's the accommodations. The hotel ranged, but you know what? Guess what? That one was probably $5 a night. So that's kind of what you get. You kind of, when at the end of the day, you're just looking for a place to sleep and you don't have a lot of choice. That's a very common taxi, public transportation that you see and have to contend with in some of these little towns. This is Amazonas, Peru, big, high waterfalls, really cool. It's not Angel Falls in Venezuela, but it was still pretty nice. This is also Amazonas. Think of sort of Machu Picchu, but this is sort of another area, pre-Columbian settlement in Amazonas. This is actually an artist's rendering of what it would have looked at when people were living there. And this is now what's coming up is what's left. You can still see the circular walls. It's taken a little, there we're loading. This is what's left. There is a lot of restoration or I should say conservation efforts taking place there. This is an area called Chan Chan. This is very close to the ocean. We've gone down to the coast. There are no trees, nothing to build with. So they did everything with mud. And in fact, you saw the shelter over that. That's what they're doing to preserve it, because even though it doesn't rain very often, it does rain and it kind of wants to dissolve. This is actually a surprise we found, a beer pub on the way to Huascaran National Park. And some examples of what they had. I really enjoyed that ginger pale ale. That was very good. This then is a picture of one of the glacier lakes in the national parks coming up. I'm seeing it. There we go. We're riding all the way up and just got this beautiful view. That's a natural lake. It's not dammed. So you see that typical green of a glacier lake. The road you see to the right, that's the road we had just come up to get to this viewpoint, this vantage point for me to take that picture. I said that hotels are kind of hard to come by, or sometimes you take what you can get. Well, sometimes you can't even find a hotel. This Peruvian family put us up in their barn one night. And I don't speak Spanish. My friend Paul, who's standing behind the young man, Paul was the guy that I went riding with. He speaks a little bit of Spanish. And he got us in and they put us up and they fed us and it was amazing. This is the world's largest copper mine. We just happened to stumble across it. We got some pretty good altitude here. There's some, they got 3 or 4 different breeds of these high mountain creatures, alpacas or llamas or something like that. We saw quite a few of those. This is a view. It just shows you how desolate it is. We're probably up at around 13 or 14,000 feet here. Just another gorgeous view. What you're looking at here, that's the Nazca plane. We're up in a stand. You can rent you can get a ride on an airplane that'll take you up to look at the Nazca tracks also. That was very interesting. The coast of South America is mostly desert, even though you got all this water to the left. When you see green there, it's because they've taken rivers. The way the airflow works is the ground heats up, it pulls moisture in off the ocean, but that won't condense and fall as rain until it's gone fairly far inland, and then it comes back as moisture. The food again, hey waiter, what's this chicken foot doing in my soup? That was when I kind of lost interest in that soup. But you know what? That's apparently very common. You can go and buy packages of chicken feed at the grocery store. I saw them. Every time I saw a hole in the sidewalk in one of these towns, I thought of Jim Skinner because of the accessibility and hazards. The hazards are just everywhere in terms of the infrastructure. It's surprising. So. Was it jungle? No. See, there's desert. There's obviously snow in the background. So again, we got into some high altitude riding. Snow right there. This is the Atacama Desert. You see me using this as my background on the Zoom presentation. Hand coming out of the desert, waving hello. And there's me looking like I need a barber. Then the next one, another border crossing. And this one was interesting. There's actually 2 dogs on that motorcycle. The big one you can see obviously, but there's a little brown one with his head. You see the back of his head. That was at a border. You see interesting things like that at border crossings. And here I am, Ushuaia, Argentina. That's as far south as you can possibly go unless you go over to Antarctica. So we made it, like I said, close to 50,000 miles on the motorcycle by the time I got there. And this is a celebration. I'm with my riding friend, Paul. The guy in the blue sweater, he was on the boat with me when we crossed from Panama to Colombia. The other guy's a German. Lots of Germans do this. I got a question, what kind of bike? It was a Kawasaki KLR 650. You bump into a lot of other motorcyclists. This is a popular thing to do with Europeans especially. You run into a lot more Europeans than English. But the guy in the blue sweater, he and a friend actually bought new motorcycles in Alaska and rode down from Alaska in through Central America, and they were both on that boat. But they both decided to split up when they got to Colombia because they were really kind of looking for doing different things. The other guy, the German in that picture, he works for one of the car companies over there, and they give them nice long breaks where they can do stuff like this. So there we go. I'll tell you what I'll do is I'll hang out after Oasis is officially over. If anyone has any questions or comments and wants to talk about this, I'll stay on the Zoom call. I hope I didn't take too long.
Oh,
Thank you, everybody.
No, it was awesome. That was awesome. Can everybody hear me
Cool.
now? Sorry about that technical difficulty earlier. That is amazing. I have so many questions, but we don't have time for it. So like Gary offered, he'll hang out after the end of our gathering. Once Mark sings our last song, you can stay here in the main lobby and chat with Gary, ask him some questions. So man, that yeah, like the amount of questions that are running around in my head are just innumerable, but yeah, we'll get to those. We'll get to those after gathering's over. So thank you so much for presenting that. And thank you to your son, Tyler, also for helping with your slides. Thanks, Tyler, appreciate it. So it sounds like I had audio issues. No one could hear me. So I want to restate the, okay, good now. Restate the celebrations. So we want to wish Lynn Thompson a happy birthday from this past Thursday. Megan Lawrence also has a birthday coming up this coming Friday. And we'd like to wish Lisa and Kevin Phillips a very happy 11th anniversary today. So let's give everybody a round of applause. Fantastic. And also Judy Bishop mentioned in the chat that her grandchildren both graduated recently and were both members of National Honor Society. So that represents a lot of hard work. And so our congratulations go out to them as well. Okay, so at this time we are going to jump into our breakout rooms, and maybe if you win the lottery, you're going to end up with Gary and his son in your breakout room. You might get a preview of some of what he has to talk about. So we'll be in these breakout rooms for about 10 minutes, and then we'll all come back together and get to listen to Mark perform again. So we'll see you back in 10 minutes. Okay. Hey, babe, how are you this morning?
I'm good.
Good. I got to quit talking. So, oh, this is still on the live stream. Did you get a room? A breakout room?
Yeah.
Okay. I'm going to mute myself. I'll see you.
All right.
Hello again, everyone. Welcome back from the breakout rooms. Hopefully you were able to catch up with someone you maybe haven't seen for a little bit of time, get a chance to chat this morning. We know that our coffee breaks are one of our favorite things when we're together in person to get to really connect. So we're happy that this feature exists so we can get a little taste of that here at our Sunday gathering. So with that, I'm going to talk a little bit more about Mark. Let's see, Mark, are you about ready to go? So Mark, like you mentioned, is part of a band called Fire Dog. And he said that really the best way to see where they have upcoming performances or to listen to their music or to hear more about them is on their website, which is See Fire Dog Roll. So it's SEE Fire Dog Roll, all one word, ROLL, .com. And that's where you can see all things Fire Dog and Mark Pagano. So Mark, whenever you're ready.
Okay, thank you.
Uh-huh.
Thank you so much. Yeah, thanks for having me. I was just telling the breakout group, it was really nice seeing Gary's presentation. I got to spend some time in Peru several years ago, so I was just kind of bringing back some of those memories of the people and the landscapes. It's a pretty neat experience to have. I'm going to play a song that I wrote after I took a trip down to the Everglades in Florida a few years ago, and I was listening to a lot of lectures by Alan Watts at the time, kind of about the nature of reality and stuff like that. So this is called Everglades. You can believe what you want to believe. All the dark energy that's surrounded by stars. I can feel the longing, all the space in between. The places we came from, the ways that we are. From the forest floor to the canopy. Walking in the shadows of the mystery, but it's all in your mind, all in your, it's all in your mind, all in your, it's all in your mind. Through the rhythms of time, each seed in its shell. The villain, the hero, the stories we tell. Where the fresh waters meet the sea. In the dark matter of the mystery, but it's all in your mind, all in your, it's all in your mind. Love the jazz hands. Love that. Thank you so much. Oh.
Boy, you guys, I am on the struggle bus this morning, obviously. Thank you. Thank you very much. I'm not sure what's going on with my audio today. We really appreciate that. So we're so glad that you were able to join us this morning. And again, you can see more about Mark and Fire Dog at seefiredogrroll.com. Yes, SEE, right? Fire Dog Roll, ROLL.com. Oh boy, my out loud spelling has taken a hit since 3rd grade. I'm not that great at it anymore. Okay, so now it's my pleasure to introduce our featured speaker for the day, Katie Griffin Hill. So she has worked in research and development for 20 years. Her primary interest is in developmental and reproductive toxicology. She loves to travel, run, and spoil her menagerie of pets, including a retired laboratory cat named Charmander. And we talked a little bit about him, and she said he is just lovely and likes to do all the things and is super willing to do what people ask him to. Sounds like a great pet. So without any further ado, let's welcome Katie.
All right, is it on the screen?
Yep, looks great.
Awesome. So, well, hello everyone. I am, my name is Katie Hill, and I work with Nikki, or did work with Nikki. She has left us for different pastures, and we're sad to see her go, but we're excited for her new adventures. I've worked in the facility I work in for about 20 years, and we have a lot of people come and go, so try not to be too sad about it. One of the, Nikki had asked me to speak today to just kind of go over drug development, because with the COVID situation, we're hearing a lot about it. Now, some of the things that are being done with the COVID drugs are a little different than what is the typical procedure because of the situation. But one of the reasons that we're seeing drugs go into humans a lot faster is because a lot of these drugs that you're hearing them discuss are being repurposed. They've already had some development done for another reason, and they are trying to basically just change the trajectory of the testing. So they're still following the proper channels, it's just expedited in this case. So normally the drug development process is rather slow and tedious. So hopefully my presentation won't be, although it definitely is not nearly as colorful and fun as Gary's, but I hope that it will be informative. So basically, new drug development is an extensive process. The FDA requires any drug that is given to humans be tested. There are very, very few exceptions to this rule, but generally we have to do preclinical development before we do the clinical development before we go onto the market. So a new drug takes millions of dollars. There's multiple statistics, but this was $350 million over 12 years. A drug that I personally have worked on for 14 years, well, 13 years, just went onto the market. We first dosed it into a rat in 2006, and it was approved by the FDA in December of last year. This is a drug for schizophrenia. Now, a drug like that is a little different from some of the drugs you would see for COVID, because most drugs for schizophrenia or chronic conditions are intended to be taken for long periods of time. So for these long periods of time, we need to test for longer in order to dose in humans for, a person with schizophrenia could theoretically take this drug for 10 years. So we have to test in animals for up to 2 years. So it's just the dosing alone takes longer. So as I said, the research and development involves two primary phases: preclinical development, which is the area Nikki and I work in. And basically preclinical is a slightly PC way of saying animal testing. And I, in high school, was completely against animal testing. And then I discovered that one, it's necessary, and two, it's the law. The biggest thing is you basically can't dose a human until we've dosed animals, because that is, at this point, the only way, the best way to guarantee safety going into people. We are working very hard as a drug development community to do what they call in silico, which is computer modeled testing, as well as in vitro, which is where we use cells. However, we have not been able to emulate the full complex systems that we can replicate with animals. So, and the animals that are used in preclinical testing, like I mentioned, I have a retired laboratory cat. They are all what we call purpose bred. All of the animals that we use for our testing are bred at facilities that are designed, that produce animals for this purpose. Research facilities do not use stray animals. They don't want, we need animals that are extremely healthy and well cared for prior to use. I mean, even my pets, I have 2 dogs that are spoiled and I like to think are well taken care of. Neither of them are even what we would call good enough for our lab. We need animals that are young, healthy, and have been what we call barrier bred. Basically, these animals are so far removed from, you know, their household origins, so to speak, that there are no pathogens that they are exposed to. So they're very healthy and specifically used for this purpose. A lot of companies are trying to go towards adopting out animals that can be adopted out after the studies are completed. And so I have my retired laboratory cat, and I also have a rat. Some people adopt out dogs, rabbits, guinea pigs, hamsters. Some tests don't allow for that for various reasons, but when it's possible, we try to do so. So once we've tested in the animals, we move into humans. And that's the clinical phase. That's when you hear the ads on the radio saying, if you are a young, healthy, non-smoking male, ages 20 to 40, you can make $1,000. That's phase 1 of clinical trials. So when we're developing drugs, when they start testing, it's a success rate of about 0.1%, 1 in 1,000 drugs, which in a way is kind of incredible that any drugs make it to market because of the stringent criteria. So as I said, the FDA governs our research. So agents or companies that do drug development work closely with them and help develop the right testing paradigms for the particular drug. For example, a drug that's going to be given once or for very short term, you know, say you're getting a steroid shot, that's going to be tested much differently than an insulin pump drug that's going to be given for possibly, you know, 30, 40 years of a person's life. So this little timeline basically shows all the complexities that go into drug development and just shows how much has to happen. There's a little arrow in the middle that says accelerated development and review, and that's what we're seeing a lot about with related to the COVID drugs, because basically the FDA is, if there's a drug that is being evaluated to treat COVID or a vaccine, any sort of drug that could benefit a COVID patient, they are focusing resources on that drug. They will also, the FDA works with something called Orphan Drugs, which when I first heard that term, was very confused, because I'm like, why are we testing on orphans? I was a little concerned. Orphan drugs are drugs for diseases where there is no treatment. Generally, those are diseases where there's a small number of people who have the condition. There's not a lot of times, there's not a lot of money in it. You know, if there's only 1,000 people in the world that have a condition, there's not a lot of incentive, unfortunately, for pharmaceutical companies to develop drugs for them, which is very unfortunate. But you know, it is for-profit industry. So the NIH, the National Institute of Health, which is a government agency, actually provides grants as well as accelerated development and review for those drugs, so we can try to get them into humans a little faster. Some other countries have procedures slightly different than ours as well. South Africa has something called compassionate use. Basically, if you have a condition that's terminal and you can get access to a experimental pharmaceutical, it's legal to use it. There's been discussions of improving that in the US, but as for now, the FDA is still, they will allow experimental drugs to go into humans faster when it's a terminal condition, but they still require some animal testing prior to that. As I said, preclinical studies are the area I work in, and that basically allows us to figure out as much as we can about the drug. We look at what we call the pharmacology. We dose a particular breed of animal, and then we take blood and sometimes tissue samples, sometimes urine samples, sometimes liver bile, and test that for how much drug is in it. Basically, we're looking for what we call ADME, ADME, absorption, distribution, metabolism, and excretion. We want to see where in the body does that drug go, how is it released. It's very important to know if the drug stays in the body in the tissues, because then you're more likely to have an overdose type situation of having the too much drug in your body. We also don't want a drug that's excreted too fast if it's something you're supposed to take every day. So there's a lot that can be done to change the way a drug is absorbed and excreted, and a lot of time is spent on that. We look at pharmacodynamics, basically how does the drug work, and does it work? We have something called knockout mice, which are mice that have had some sort of gene altered or knocked out that allows us to model a disease. You can purchase mice that basically have the mouse version of cystic fibrosis, and you can test for the drug to see if it actually works in mice. That doesn't guarantee that it will work in humans with cystic fibrosis, unfortunately. My area of expertise is toxicology, and my particular favorite is, like I said, developmental and reproductive toxicology. That's basically when you give a drug to either a mating pair of rodents or to a pregnant female rodent, and see if there's effects on either the fetus or the offspring. It's very interesting, and it's one of my particular, like I said, areas of interest, and it's also very important because we don't have the ability to test on pregnant women the same way we do healthy adults. If you're a 25-year-old male, we'll give you pretty much any drug that you're willing to sign up for in phase 1, and we don't do that in pregnant women the same way, because it's basically considered unethical. There are some exceptions. If a pregnant woman is taking a drug and becomes pregnant, obviously sometimes it is not in the best interest of the woman to stop taking it. You know, if you have a woman with mental health issues that requires daily medication, but in general, drugs are not prescribed to pregnant women as readily as they are women who are not pregnant, because we don't have the human safety testing to back it up in all instances. It's really more of a case-by-case basis. These studies hopefully will establish the safety of the drug. Basically, we determine what dose is considered safe in animals. We take that down tenfold, and then we can introduce it into humans. So basically, once we have tested in 2 species, one rodent, one non-rodent. Fun fact, I did not know rabbits were not rodents until I started working in this industry. Generally, the gold standard is that we test in rats and dogs. It's beagle dogs generally, but there are some exceptions for large molecules such as proteins or antibodies. A lot of times we'll test in monkeys. Our particular facility tests in crab-eating macaques, which are about, they're little guys, about 2 to 4 kilos. Super cute, although very bitey, as are mice, also very bitey, but tiny, so it's nice. So for rodents, basically you're looking at rats or mice. For the large animals, generally it is dogs, primates, or what we call miniature swine or mini pigs, which our facility didn't have mini pigs when I first started. And then they said we were getting, sorry about that. They said we were getting mini pigs. I thought we were getting teacup pigs, like cute little tiny pigs. No, they're like 50 pounds. It was really disappointing, but they are smaller than production swine, and they are helpful for, especially for skin models and the cardiothoracic drugs. So basically, once we've done these 2 tests in these 2 species, the company can go to the FDA and present their what they call IND or investigational new drug package. Once that is approved by the FDA, then they can go into humans. As I said, their phase 1 is the healthy volunteers, and then we move into testing into phase 2, which is where we test in actual patients. And then phase 3, larger patients, then hopefully you can go on to regulatory review. And then phase 4, we actually go on to market. Phase 1 is, like I said, the safety in healthy volunteers, and a pretty good chunk of drugs that make it to this phase will pass. Because generally we've established safety. Like I said, we do a 10-fold lower dose. So hopefully when we go into humans that have no background health issues, we'll see no significant issues. Then we move into phase 2, which is the efficacy testing. And you hope that we establish efficacy in the mouse models or in vitro testing. However, until we get into phase 2, we really don't know if it works. And so about a third of drugs make it through this phase. This is a really big deal for drugs for mental health because it's much harder to quantify. A lot of the efficacy of mental health drugs is really based on a bit of a perception. And I worked on the drug that I worked on that I said was approved in December 2019. They had to do several phase 2 and 3 tests extra because of the placebo effect. They saw that patients receiving the placebo actually reported an improvement in symptoms, and the drug needs to be better than the placebo because it's not worth the side effects otherwise. So about a third of drugs that make it to this point, then will make it through there. Finally, we go into phase 3, which is again in patients, but again in diseased patients, and about a third of drugs make it through this phase. We submit a new drug application at this point, and then the FDA will decide. It says 60 days, but generally for the COVID drugs, it's going to be much faster. So the FDA has responsibilities to the citizens to basically determine that the drug is safe. And they also inspect where it's manufactured to make sure it's being made properly. But then ultimately, it falls onto the sponsors or the manufacturers of the drug to continue to prove safety and maintain data on what they call adverse events or AEs. If a person takes a drug and goes into anaphylactic shock, they need to notify the doctors that treat that patient will notify the manufacturers and the FDA so that that sort of situation can be monitored. The last phase is phase 4, which is the drugs we take are in phase 4. for quite a period of time, depending upon the type of drug and the number of people taking it, as well as the potential for adverse events. And that is it. So hopefully that was educational and of interest. So I'm open for questions if you have any, and
Okay.
if we have time.
Oh yeah, absolutely. So I have a few here in the chat, and people can please keep adding to that as we go. So this is from Nikki, and she talked about, and you maybe talked about the different preclinical stages, but then she threw me an acronym, which I don't know what it means. So I'm going to go ahead and read the question anyway. She said, what are the different preclinical stages, i.e. non-GLP versus GLP?
So GLP is, the term is good laboratory practice. There's actually two other terms that are similar, which is good manufacturing practice, or GMP, and good clinical practices, which is GCP. All three of these sets of regulations govern clinical, preclinical, and manufacturing of drugs.
Okay.
Basically, it is rules regarding documentation and procedures to ensure that all of the work that is done can be replicated if needed. Basically, if I run a study and I say, when I give this drug to dogs, blah, blah, blah happened. Well, someone else needs to be able to do that and make it happen if needed. It's basically replicating or reconstructing what was done. This way we ensure, basically it's a set of regulations that ensure, quote unquote, ensure good science. It's not 100% what it does, because you can do procedures poorly and record them properly, but that's what the goal is.
Yeah. So when
Cats,
you do,
oh, go ahead.
oh, you saw the thing about cats? Yeah, what cats are
Yeah.
used
So
for? Okay.
cats actually aren't used nearly as much as other species, because as anyone who has been around pet cats or dogs, dogs are very cooperative. We use beagles. Beagles love people. And the beagles that they breed for research, I mean, they definitely breed for temperament. If a dog were to growl at someone at the breeding facility, it's not going to move on to breeding colony.
Right.
Cats, not so much. They have not bred that feature out of the, or complacency into them. Cats are generally, for the most part, only used for products that are going to be given to cats or dogs. Obviously, they're tested because they're going to go into cats, but they're also tested because clinical trials for animal vaccines and animal pharmaceuticals are tested the same way human drugs are, except for they try to use what they call the target species as much as possible. For products that are intended for dogs, a lot of times they will test them in cats at least once, because people don't always read the labels, and people say, oh, this is a flea product for dogs, well, let me put it on my cat.
Yeah.
So you want to ensure that it's safe for both. But cats are definitely used less frequently than a lot of the other species, and than dogs and mice, because they're just a bit difficult to work.
That makes sense. I am a cat lady myself, and yeah, I can barely get my cat to do anything for me, and we've had 11-year relationship, so I can't imagine him trying to cooperate with anyone else in the lab. So another question that I thought of is, so typically there's when trials are going on, are there several labs at once working on it, or is it just only after some results have been compiled and then they have to verify them, or kind of how, are there several companies working on the same drug, or is it very specific
It really
in the
depends.
development phase?
Yeah, it really depends on what, basically, is it a brand new drug? In that case, then generally there is one company, the manufacturer is the one who's focusing on developing it. However, they will frequently have other companies do research for them. The facility we work for is what they call a CRO, or Contract Research Organization.
Okay.
And that's what we do not actually produce any drugs ourselves. We do the work for other companies.
I see.
If it is a drug that is being repurposed and is already on the market for something else, and the patent has expired, then multiple companies can do the drug. An example of that is, I can't remember now, unfortunately, what the name of the drug is, but it was recently determined that basically a slight modification to the molecule meant it could be used possibly for depression. And so multiple companies were working with that.
Yeah.
And yes,
hydroxychloroquine. You saw that question.
About the, yes, they are
Not-
treated humanely, and we do a lot of vet care. I agree. I was anti-animal testing for a long time. These animals live very good lives. They are, every test that is done in a facility that uses animals has to be approved by an animal welfare committee. And we have multiple procedures in place regarding humane endpoints. We do not allow animals to suffer, because that's not good science for us either. And a lot of the people are animal lovers. A lot of us have our own pets. And you can't do the work we do without caring about animals. It's just not really feasible. It's especially the people who work day to day with the animals. We have a lot of people who've worked in the vet tech industry. And it's definitely not fun when animals have to be euthanized, but these are legal requirements in order to get drugs into humans. And so we do everything we can to make their lives good while they are helping us. And really, I mean, like my dogs, they generally eat when I get up. If it's at 6, they eat at 6. If it's at 9, it's at 9. The dogs at our work get fed on a much tighter schedule. And the thing is, these animals, like I said, they're all barrier captive bred. They don't know any better. So they think their lives are pretty good. It's not like they've been out in someone's home and then were brought to the lab. Now, obviously, if they're adopted, like my cat, yes, he probably has his life here is better because he has a whole house to roam in. But he didn't know any better until he left. So we care for them as best we can, and they, keep them as happy and healthy as can be, so that they help us help society. And there was a question about skipping phases if a drug is already on the market, and that is correct.
Right.
Yes. If the safety testing has already been done, or if they're only modifying a drug slightly, this happens a lot with oral medications, is the manufacturer will make some changes to the pill or capsule that they're doing, and they can usually get by in that case with very minimal or no additional animal testing. They can usually, a lot of times what they can do is do a small study where they take the new formulation and the old formulation and basically compare them side by side, and as long as the new formulation isn't worse for the animals, they can go into humans much quicker. And that's what they can do with COVID. Compare your procedures to those of Europe. Europe is much more conservative in terms of animal use. And there are instances where so Europe is basically governed by the OECD. The OECD does GLP a little differently. They inspect every facility and certify them, whereas the FDA comes to the facilities on a either 2 to 3 year basis and or as needed. Like basically any company that's working on a COVID drug, if that drug gets approved, the FDA is coming to your facility to look at your records. In general, Europe is more conservative about approvals as well. But for the most part, if the FDA approves, the OECD is likely to do so, and vice versa, because the rules are pretty similar. They will generally, it's not exactly a like for like, oh, if the FDA says yes, the OECD automatically does, but a lot of times they will approve, both agencies will approve the drugs eventually.
Okay. All right. Does anyone else have any questions? Go ahead and put them in the chat. That was a lot to think about. And I know, of course, this is a very top of mind discussion for everyone right now in light of what we're all going through with the pandemic. I'm sure people just in your life in general were ask you this question, like, how close are we to getting a vaccine, for COVID-19? I think that's probably something that's really on everybody's mind. So it's really interesting to hear, all the process that it takes and the time frame that goes with it. Oh, Nikki wants to know if the study she worked on was approved in December.
I don't think so, because I think the last time we worked on the drug was a couple years ago. And I think it was before your time. It was, the company's name is Intracellular Therapeutics, so ITI. And I honestly don't know what the market name of the drug is. We called it ITI-007, but it was approved and it's being marketed as something else. So I am certain that you have worked on things that will eventually make it to market. Just got to be patient.
Yeah. So that was another question that kind of came to my mind. Like in your career, do you have an idea of how many drugs you've worked on that have actually made it through all the phases?
That is an interesting question. Our facility actually didn't do drug development. We did pesticide and EPA testing until 2006.
Ah, okay.
So, and the area I worked in was developmental neurotoxicology, and a lot of the pesticides and insecticides and other agricultural chemicals that I worked on were actually already on the market. The EPA is very particular, and they basically, if neurotoxicology testing wasn't actually implemented until the late 80s, early 90s. So basically what the EPA does is if they come up with new testing, they send a letter to companies that make the agricultural products called a DCI, or data call-in notice. And they say, hey, we came up with this new test. You need to do it or your pesticides off the market. I mean, it's nicely worded, but basically that's what it is. They did not have developmental neurotoxicology testing until the mid-90s. So they test on adults came up in the, I take it back. Testing for adults came up in the late 80s. Testing for developmental neurotox came up in the mid-90s.
Wow.
And so basically drugs where they felt, pesticides, insecticides, et cetera, where they felt there was a neurotox risk, the companies were required to do this additional testing. So the EPA is extremely good about going back and asking companies for additional research. Also, if a college or some sort of other institution does testing that shows, like, basically if you hear about, bad water, contaminated water, the EPA will require companies to do additional research if it shows up that, so I personally worked on a study where trichloroethylene was found on in the water on military bases in Texas. Trichloroethylene has a lot of uses. One of the most common is as a dry cleaning solvent. And so we did a water study, basically giving pregnant female rats treated water to see if there were effects. And now in this case, because it was found in the water, there's only so much that can be done about it from that regard for preclinical testing. but at least we were able to show that at certain doses, what dose level it's safe at, and they could determine if the water is quote unquote too contaminated.
Yeah. I grew up in a central Washington state during the whole ALAR sort of, when they were using that as a pesticide, and now have sort of, the spray wagons and tractors would come right past us, and we'd get spray on us on our bus at the bus stop. So that explains a lot, y'all. If you want to know what's wrong with me, it may be that, because I know since then they've done some tests about that. Someone's asking, what is the status of Roundup?
I actually do not know. I know that's been a hot button issue, but I personally am not following it all that closely. I don't know if anyone else knows.
Yeah, well, we can Google it up maybe later on. Okay. Well, we appreciate so much, Katie, you taking your time with us this morning and this afternoon to share what you're up to and give us some insight into, how these preclinical and clinical phases are developed. It was really fascinating. So we appreciate it. Thank you so much.
Well, thank you for letting me join you. I have enjoyed the last few weeks of Zooming in after he introduced me to Oasis. So thank you.
Thank you. Okay, so at this time, I'll just remind everyone that Oasis is a 501c3. We're a nonprofit organization, entirely volunteer staffed and donation funded. And our program today is just part of the wide range of informational, educational, and motivational speakers we offer most Sundays. So if you appreciated what you've seen and heard, please consider making a donation. There are several ways to donate. There is text to give. The number for that is 816-839-7656. With the dollar amount that you'd like to give, you can text that. And then online, you can go to kcoasis.org and click on the donate button at the top of the homepage, and that will give you the instructions of how you can do that. So with that, we will go back to Mark for our final musical performance, and then let everyone get on to their rest of their Memorial Day weekend. For those of you that have military background, for any of those of us that have lost people due to their participation in the military, we want to keep those people in our thoughts, and we appreciate the efforts of all of our military folks. So with that, Mark, we'll go ahead and have you take it away. Again, you can learn about all things Fire Dog and Mark at CSEE firedogrol, ROLL.com. And we're excited to hear your last tune.
Oops. Hello? Can you hear me now? Yeah, okay. All right, thank you. Thanks so much. Thank you so much for having me. I'm glad to connect with you in this way. I've just been, like so many of us, kind of taking inventory of the things around my house and thinking about my song catalog, and I came across this song that I wrote several years ago. So I'm going to end with this one. It's called Time Bomb. How long I wish we'd won, but now we're under the gun. Taking my time, it's just another deadline. You don't know how it's gonna go. But readiness is building, we're bound to break the cage. My heart is fluttering amid the winds of change. But I know we're gonna make it, though things won't be the same. We're gonna find a better way. Cause readiness is building, we're bound to break the cage. My heart is fluttering amid the winds of change. But I know we're gonna make it, though things won't be the same. We're gonna find a better way. Readiness is building, we're bound to break the cage. My heart is fluttering amid the winds of change. But I know we're gonna make it, though things won't be the same. We're gonna find a better way. We're gonna find a better way. Thank you. Thanks so much for having
Yay!
me
Thank you. Thank you.
in the future.
Yep. And Mark, I hope that sometime in our future we'll cross paths and you'll get to join us here in person in Kansas City again sometime. So thank you. Thank you so much.
Thank you. Have a great
Okay.
weekend, everybody.
Thank you. And with that, I'd like to thank all of our presenters, Mark Pagano, Gary Blakely, Katie Griffin Hill. And I just want to give everyone a reminder that throughout the week, we have other opportunities for you to meet up with your fellow Oasis folks using this exact same Zoom meeting link. We have a coffee talk at 7:30 a.m. every morning, and then also on Wednesday evenings at 8 p.m. So with that, we're going to go ahead and stop the live stream, and I will see you all you lovely, wonderful Oasans this time next week. Until then, take care.